Network Entertainment.
You think of it as like, women, if we want to save our eggs, we put them in cryobanks, right? If we want to get pregnant later, it's that idea that we will freeze our eggs, and then if we want to get pregnant, then we will get the eggs later if there's an available sperm that we like. This is the fourth species that was cryopreserved. So the first one, so it's a larvae from a spawning coral, a mushroom coral. They were able to cryopreserve the larvae, but at just cryopreserving and then thawing it, and then it survived for a couple of hours or days. Second and third species, it was still mobile after thawing, but it didn't settle and grow to a adult coral. So for me, so this is why it's the first one. It's not the first one that the larvae has been cryopreserved, but it's the first one that it was cryopreserved, thaw, it settled, and it grew.
Sa tuwing nagbabasa kayo ng mga research papers, nagtataka din ba kayo kung paano ito naisipan gawin ng mga researchers? Paano nila na-transform ang idea nila para maging isang actual na research project? Saan sila kumuha ng funding para magawa to? Paano nila nalaman na ang results nila ay pwede na nilang ipasa for publication? Dito, sa Behind the Science podcast, ay kakausapin natin ang ating mga Pinoy scientists para ibahagi hindi lang ang mga resulta ng kanilang pag-aaral, kundi pati na rin ang mga behind the scenes or behind the science stories nito. Simula sa kung paano nila ito naisipan gawin hanggang sa na-publish na nila ito. So this podcast is in partnership with the UP Resilience Institute, the Philippine Agricultural Scientist, the Philippine Journal of Fisheries, and SciEnggJ. So ang UP Resilience Institute ay long-time partners na namin dito sa podcast. Nung nagsisimula pa lang kami, nandyan na sila para tumulong sa amin. At para lang sa kaalaman ng lahat, ako ngayon ay isang UPRI Research Fellow. The Philippine Agricultural Scientist is the first international journal in the Philippines and Southeast Asia in the field of agriculture. It is published by the University of the Philippines, Los Baños, College of Agriculture and Food Science. It publishes articles in the fields of tropical agricultural science and related areas including environmental science, food science, engineering, biotechnology, economics, rural sociology, development communication, agroforestry and silviculture, and marine and fishery science. The Philippine Journal of Fisheries is the official publication of the Department of Agriculture, National Fisheries Research and Development Institute. It covers all aspects of fisheries, both in basic and applied sciences, as well as related fields of studies such as capture fisheries, aquaculture, post-harvest technology, marketing and industry, ecology, oceanography, conservation, biotechnology, food safety, socioeconomics, and systematics in freshwater, estuarine, marine, and other identifiable water habitats. SciEnggJ, formerly known as the Philippine Science Letters, is the official journal of the Philippine American Academy of Science and Engineering. It covers studies in the natural, physical, mathematical, computational, and social sciences, including those related to agriculture, health, engineering, environment, education, and technology. So kung may paper kayong gustong i-publish at pasok ito sa scope ng mga journals na ito, please consider submitting to one of them. At syempre importante, take note na hindi kayo pwede mag-submit ng sabay-sabay sa tatlong journals na ito. Kasi bawal yon. Dapat sa isang journal muna kayo mag-submit. At ang mga journals na to ay index na rin sa Web of Science, Scopus, ASEAN Citation Index, and SciEnggJ Journal Ranking. Kaya masisiguro nyo dito ang mataas na standard at mabusisi na review process para masiguro ang high quality papers na pinapublish. So if you're interested in publishing your research in one of these journals, check nyo ang guidelines at ang links ay nasa description. Magandang araw mga kapwa-scientista at welcome sa isa na namang episode ng Behind the Science podcast. Ako si Paul Flores, ang inyong host, and our guest is a postdoctoral associate at the Arabian Center for Climate and Environmental Sciences, New York University, Abu Dhabi. She earned her PhD in marine science from National Sun Yat-sen University in Taiwan, where her dissertation focused on the cryopreservation and cryobanking of coral larvae. She also holds an MS in marine science, major in marine biology from UP Diliman, and a BS in Marine Biology from Xavier University, Ateneo de Cagayan, where she graduated with the Best Thesis Award and recognition as Most Outstanding Student. So without further ado, let's all welcome Dr. Arah Narida.
Hi, Paul. Good afternoon. Thank you for inviting me to your podcast.
Ayun, so welcome Ara sa podcast. At bago pala tayo magsimula, no, kung nag enjoy kayo dito sa podcast at gusto nyo ang ginagawa namin dito, paki-like naman ang episode na ito at mag-subscribe na rin kayo sa mga social media platforms ng Behind the Science Podcast. Makikita nyo kami sa Spotify, YouTube, Apple Podcast, and Facebook. Paki-rate na rin kami ng 5 stars para mas madami pa tayong maabot na audience. So ang topic natin today, actually, ito yung naging PhD dissertation ni Ara tungkol ito sa cryopreservation or ang pag-freeze ng coral larva at kumbaga defrost sa kanila para mag-grow into adults. And dito sa podcast, madami na rin tayong naging episodes on corals and coral reefs. So check nyo lang ang episode list namin. Pero yung mga latest episodes tungkol dito sa coral reefs ay episode 83. Ang pinag-usapan namin ay yung mga challenges sa regulation ng coral reef restoration projects sa Pilipinas with Mr. Erlo Matores, and episode 69 tungkol naman sa bagong grading system para sukatin ang health ng isang coral reef with Dr. Wilfredo Licuanan. So ayun, bago natin pag-usapan yung yung research ni Ara, siguro mas kilalanin muna natin siya as well as yung naging journey niya as a scientist. So Ara, yung first question namin dito palagi sa podcast, kailan mo na-realize na gusto mo magtrabaho sa science sector?
It started when I think from my memory, it started when, like, during high school. So it's, when you see underwater videos and like, oh, they're so nice, they're so beautiful, the fish, the coral reefs. So I told myself, like, I want to be a marine biologist. But I didn't know at that time that there's a marine biology course in Cagayan de Oro. So I just want to be a marine biologist. And then when I graduated high school, I applied for Xavier University. And then actually I applied for food tech, food technology, and something else. But then I got in, and then I end up doing marine biology. So at that time, like, oh, I want to be a marine biologist. I want to do marine biology because I want to dive. And then after that, I was like, when I got my certificate, I like my diver's license, I said, like, maybe I should shift because, like, I have this thing that, like, oh, I've already done this, and then maybe I'll just change course. But it turned out like I loved it because it's like marine biology. It's for me. It's there's a lot of not field work. Like, we do it's hard, but at the same time you get to go out, you get to swim. Like, it's more like a field trip every time we go out. So, and then we go back to the lab. So I finished my degree, my BS, and then I told myself, because I don't know what to do. I always have this thing, like, I don't know what to do with my life. Like, I am a type of person that, like in the moment, like, I don't really plan ahead, but like, I told myself before my defense that if I get the best thesis paper, then I might go to grad school. And true enough, I got the best thesis award. So I said, like, okay, maybe I will go to grad school. So my first job was not actually um, in the marine biology field, but as an admin assistant at Xavier University. So, but it was under the research of like research center of Xavier. So I got that job, which could be a plantilla position. So after 6 months, I will be a regular employee of Xavier. But before I submitted because I was on probation for a few months, before I submitted my application for that position, my first boss told me, which is Dr. Hailey Kawit, she told me that she has a project on giant clams. But this position is temporary. It's by contract, so it's 1 year. But I told her, like, because I told myself, like, I really want to do grad school, so like I'll just forego this admin thing. And then she even told me that the salary is even lower. So like I don't care because I want to do grad school. So I didn't submitted my application, and then I transitioned to my first marine biology job with a giant clam project. And then it turns out also, like, because it was a contractual um job, so that at that time the salary was higher compared to uh
Ah,
the position that the admin assistant, but it turned out that way. So I worked for 2 years for a giant clam project in Cagayan de Oro. So the goal at that time was to, because the giant clam population is declining, so we want to restock the giant clams in northern Mindanao. So that was my first project. And then after 2 years, Doc Hiley told us, like the research assistant, not just me, that you need to leave. You need to leave because so that your career will also progress. And then it's at that time there was a new project with UPMSI, which is the integrated coastal and is coast. I forgot, it's called ice cream at that time, but it's more about looking at resilience of corals. So that's why I ended up in UP. So my second job, so before that transition, I had two offers. One is for plankton and the other one is for corals. So I was like, what should I pick? Because like there's two jobs available for me. And then so I consulted my boss and I told her, like, what should I do? So she told me, like, you choose something that will be your PhD so that you will not start again. So it's not a hard transition from master's to PhD if you want to do PhD. And then at that time also I was, because I did my undergrad thesis was also on plankton. That's why I applied for the plankton position. And then so I chose corals. So I was like, I don't want to do the lab work anymore. Like, like looking at microscopic things, just be in the lab, looking those small plankton. So I chose the coral path. So I did my coral work for I started my so that was so corals was I did my core, like pure coral ecology and taxonomy for from 2009 to 2020.
ah.
And then COVID happened, and then, like, what do I want to do with my life? Like, I don't know if I still want to do PhD, but because of COVID, we cannot go out. And then my job is so dependent on going out, diving, collecting data, because I was doing consultancy work too for companies. So I can't go out. And then like, I'm lucky enough that there was a lab available, but it was not it's not coral ecology, but this is the where the cryopreservation came in. So there was an opportunity for PhD with it's a little bit different to what I have been doing for the past um 10-15 years of my career. So that's why I ended up doing cryopreservation, but still on corals, but in a different approach. So it's more technique. So that's like my a little bit of my history with marine biology, like how I ended up here where I am right now. And then now I'm going back. So from cryopreservation, now I'm back again to coral ecology, coral reproduction, and coral spawning, looking at thermal tolerance of corals in the world's warmest sea, which is Abu Dhabi.
Ah. Nice. Yung ano po pala, siguro balikan ko lang po yung BS Marine Biology. Hindi gano'n kadami yung schools na nag-o-offer nun sa Pilipinas, 'di ba? Kasi within UP system wala kang BS
No,
Marine Biology, eh.
no. So from what I remember, Xavier University was the first university who offered marine biology in the Philippines. But we don't have, from what I know, we don't have enough teachers to like grow the, like, compared to UP. Like, we have really the Research Center, Marine Science Institute. So w- we only have undergrad. So at that time, during my time, so that's why they call us we have the few, the proud. Because during my time, first year to fourth year, we were only 16. 16
Ah,
students. First year to fourth year, can you imagine? So in my batch, we graduated 5 people. There's 5 people. So it's such a small community. S-
gra
So
gano'n lang pala kakaunti.
so there's not, so it's so ironic that we are an archipelagic country,
Oo,
but there's not many people who study marine biology in the
oo.
Philippines. So every time people will ask me, like, what do you do, or what's your course? Marine biology. They will think of maritime, or like, do you want to be on like the sailors? What do you call them?
Yung parang seaman gano'n?
Seaman. So you will be, no, 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 no. It's not that. It's like studying biology, but for the marine organism. So I still need to explain myself, even to my family, that, oh, this is what I do. It's not going to boats and being a sea woman, or yeah, seaman. So it's not that kind. So yeah, there's a few, because I think also, like for Marine Science Institute, they're thinking of the founder before is like, they don't offer marine biology for BS because it's too specific. So that's why I think there are only few universities in the Philippines who offer marine biology as a bachelor's degree. It's usually biology major in
Ah,
marine biology, or biology major in zoology or botany. So it's too specific. So even in UP, they have marine science, but it's only for grad school. It's not offered for bachelors.
siguro yung closest na ay yung BS Fisheries. I think
Fisheries, yes.
madaming schools nag-o-offer ng BS Fisheries.
Yes, especially for, I don't know if you have interviewed people also, like from the Visayas,
Meron
and
pa,
then there's also from, yeah, Mindanao. So fisheries, because they have a different track. They also, like, they need to take license, right, to be a
oo.
fisheries technician. Yeah. So it's not the same for marine biology. We are like, I think it's a, I don't know. It's like, if you graduated as a marine biology student, you don't have a you don't have a license. So you
Oo.
need to do further studies to be something. I think for me it's not enough. So it's like biology. It's a pre-course preparatory course to being a doctor. So that's why there's not much people. It's not very attractive also, I think.
Hmm.
Unless you go in deep into to the the study or course, that you will know that, oh, this is fun. Because, like, yeah, for me it's fun.
Ayun, siguro sa usaping attractiveness ng marine biology kasi nakuwento niyo nga po more than 10-15 years kayong nagre-research ng coral reef sa Pilipinas. Ano po yung mga napuntahan niyo at para sa'yo, ano ang pinakamagandang, site yung napuntahan mo na ano, pinakamagandang coral reef na napuntahan mo?
Okay. So, coral reef, there's a lot of, because this is a, like, if we talk about like cover, like, where are the best reefs that you can find, I I can't remember on the top of my head right now where. There's a few places in the Visayas that I've been to, that coral cover is like 90-95%. Like it's a lot because there are marine protected areas. But in terms of like um a reef that is full of is a mix of corals, large marine vertebrates is Tubataha. I think
Mm.
it's the best for me, it's the best place in the Philippines because it's not also very accessible to people. It's 10 hours away from Palawan. But you can see their sharks, mantas, whale sharks, tiger sharks, turtles, like all sorts of big things that you cannot see outside of Tubataha because it's very well protected, which I'm so proud of, like of the people who are like the Tubataha Management Office who is very much dedicated to preserving this part of the reef in the Philippines. And we also have there's also nice reefs in, like I remember before the bleaching, there's a lot of nice reefs also in Colyon. The it's near Coron, the northern part of Palawan. So we also surveyed from north of south of Palawan, and also Tawi-Tawi. Tawi-Tawi has one of the best reefs, but the current is so strong. Like it's so bad, but like it's beautiful down there. Hey, yeah, there's still we have nice reefs in the Philippines. But like the reefs near shore or the coast near the coast, they're not we all like I can say we have reefs that are nice, but most of our reefs are also in poor conditions because
Degraded.
of a lot of uh, anthropogenic um, impacts and also bleaching that's been happening all over the world, which we cannot control.
Kanina po na nasabi niyo na ano, during the pandemic may nakita kayo na opportunity to pursue yung PhD sa Taiwan. So can you share po paano niyo ba nahanap yung ganitong opportunities? Paano niyo siya saang website niyo siya nakita or may kinontak niyo ba yung supervisor niyo or something?
So the end start of pandemic, um, so I remember at that time I was diving. I think the last one was in Mindoro, and then Duterte or like our president at that time announced that we will have a lockdown. So we needed to go back. So I was stuck at home. And then I asked my friend, Dr. Maria Vanessa Barria Rodriguez from the Marine Science Institute. So because I've been emailing at that before pandemic, I've been emailing professors like, oh, I want if there's an opportunity in your lab, can I apply? I work on this kind of field, but some professors, like I don't know if you experience this, but like there are professors who will reply that,
Ah,
oh, we don't, yes, we want to have you, but we don't have funding. There are professors that will not respond, they don't have money. So I just keep maybe 10 10 or less than professors at that time. And then so I asked Van that I saw a post before, but I didn't check because I was busy on field. And then pandemic happens. I asked her like, "Oh, is it still available?" Like, because that professor came to the Philippines for a collaboration with her. So, and then she told me like, "Oh, he is looking for students." So I tried my luck. So I emailed Dr. Chashin Lin of he's connected with National Donghua University and National Museum of Marine Biology and Aquarium. So he's not connected with National Sun Yat-sen University. So I emailed him asking like, "Oh, hi, I'm Ara. I am Dr. Van's friend and I'm looking for a PhD opportunity. Is there still available in your lab?" And then he replied. So I was like, oh, okay, this is nice. So I'm excited to process things. But because it was COVID, it's so hard to get documents. It's so hard to go out, right? I don't know
Ah,
if you were you in the Philippines at that time?
ay nasa Pinas din po ako nung 2020. So,
Yes,
yeah, naghahanap din ako ng PhD opportunities nung time na yun.
it's so hard. Like getting your diploma and TOR from UP will take, I don't know, I think it took me 2-3 months.
Ah,
And like I'm emailing the registrar, but I cannot be mad at them because they cannot
ah,
also move, right? So like, oh
ah,
please, like I need my to go. And then I needed to do the medical. So I needed to go out. But going out also entails a lot of like masks and protecting yourself. And I've been exposed a couple of times while I was still in the Philippines before going to Taiwan. So it was very stressful for me because of the situation that we are in. But when I arrived in Taiwan, it was so different because at that time in Taiwan, it's like we don't, yeah, it's there's COVID, but it's normal. Like people will still gather. It was so different from the Philippines. So yeah, but the process from February 2020 to I left the Philippines September 2020. So from
ah,
Feb to August, it was
ah.
bad. Like it was so bad. If there's no pandemic at that time, it would be easier,
Ah,
right? Because like you can just go and request things and then you go to this without doing the protocol that you need to do this, you need to do that. So yeah, that I think that was the tough part of my experience applying for PhD in the Philippines. Because when I get to Taiwan, everything was like it was okay. They took care of me very much.
so since nakwento na ni Ara yung backstory sa pursue niya ng PhD, which ayun nga yung pag-usapan natin na research product ng kanyang PhD, pag-usapan na natin yon. So ang title nito ay First Successful Production of Adult Corals Derived from Cryopreserved Larvae. So Ara, can you explain muna yung paper mo? Tungkol saan ba ito in one sentence?
So my paper is about freezing a baby coral so that when you thaw it or when you warm it, it will grow into a new coral. So it's like I always tell this, like if people will ask me if you watch what's the superhero, the Chris Evans. Was it
Captain
called
America?
Captain America? Yes. Okay, yeah, it's like Captain America, right? Like he was frozen and then people found him and then treated him and then he was okay again. So it's like that. So it's like freezing the coral so to let them sleep for a very long time. And then at the when the so that's the idea. Like we want I want to make the baby sleep for a while. And then when the conditions are okay, then I will thaw it and then let it grow to be a new coral. So it's a pretty young field of science, but it's already possible right now. It's just a matter now. So the challenge now or the bottleneck now is how do we scale this up? If we will apply this to coral rehabilitation or to the conservation or coral reproduction in the future. But with our um, experiments, it is possible to freeze larvae or baby corals so that we can grow them later. So it's like, for example, there's a catastrophe, there's a bleaching, there's earthquake, there's typhoon. So we have this pool of larvae that are frozen. And then because now is not the right time for them to grow. So
Hmm.
you freeze them first and then when the conditions are good, you thaw them, and then you let them grow. It's also a good material if we want to do banking or like saving the genes of these species. If later on we want to do crossbreeds, so we have the biomaterial, we have the genes of these organisms, of these corals, f- for future experiments.
Dito po sa study nyo, may emphasis dun sa yung phrase na first successful. So siguro as a background lang din siguro sa audience, sa lahat ng nakikinig na nanonood. Ah, bakit po ba mahirap yung cryopreservation ng coral larvae?
Uh, because the material is big. So you think of it as like, women like us, to if we want to save our eggs, we put them in cryo bags, right? Like if if we want to get pregnant later, it's that idea that we will freeze our eggs, and then if we want to get pregnant, then we will get the eggs later if there's an available sperm that we like. So in cryopreservation, it's usually a sperm and egg that are cryopreserved because they are smaller. Especially sperm, it's easy to freeze them and then you can just use them later. But the challenge with larvae, one is the size, so it's big. And then there are a different compartments also that in the cell or in that tiny larvae where it will have different kinds of would I say like the ability to be frozen at that time. So different components. So that's another issue. So my experiment focused on, because this is the fourth species that was cryopreserved. So the first one, they were able, so it's a larvae from a spawning coral, mushroom coral. So they were able to cryopreserve the larvae, but at just cryopreserving and then thawing it, and then it moved for it survived for a couple of hours or days. Second and third species, it was able to um it was still mobile after thawing, but it didn't settle and grow to a adult coral. So for me, so this is why it's the first one. It's not the first one that the larvae has been cryopreserved, but it's the first one that it was cryopreserved, thaw, it settled and it grew. So, and then the tricky part of also for larvae is it's not a once like the protocol is very straightforward. So if you follow the steps, it's easy because it's almost the same. But to optimize the mix of the we call cryoprotectant, this is the solution that will protect the cell or your organism or your sample during freezing and cooling. So it's very sample specific, species and sample specific. So this is why I'm saying that in earlier in our conversation that now it's possible, but scaling up this technique, it's still a bottleneck for us because we need to optimize the technique for the organism or the species that we are working on. So it's getting there, but it still needs a lot of work to do that.
So bale before po ng study nyo, wala pang successfully na preserve niya, tapos na defrost, tapos nag-grow into adult yung larva.
Yeah. So this is the first one that it was able really to grow. Alam mo yung the when you see it, like, because this is also another, we also in my study we also employ I also have a new technique Because for the longest time, for cryopreservation, it's usually slow cooling or fast cooling. And then I am using ultra fast freezing and ultra fast cooling, ultra fast warming. So what does this mean? Because um, when you freeze something, if it's if the freezing is slow, there's a lot of ice crystal formation that will be created outside your sample and inside your sample. So we are trying to avoid that. So for my study, I used vitrification, which is a type of cryopreservation that uses ultra fast freezing and ultra fast rewarming. So what does that mean? So we have a technique now that to avoid the formation of ice crystals, we will have vitrification solution. So this is the cryoprotectant that I was saying earlier to protect the cell. So it's super fast that we can avoid the formation of ice crystals. And then and the rewarming is also super fast using lasers. So can you if you imagine like cooling the sample very fast, and then when we rewarm it, we will hit it with a laser. So I am shooting actually the baby corals with laser. But because there is a cryoprotectant that protects the baby coral, no, the larvae, so so even though the laser is so hot, it will not destroy the larvae.
Parang di siya
And
naluluto. Parang gano'n.
so it will not be cooked. So, by doing this, because it's so fast, it will not the ice crystals will not form compared to the older methods that people are using. So this is a very important technique now that we are also it's fairly new, so there's not much people doing vitrification. There are people doing vitrification for fast cooling, but there's not many people doing ultra fast rewarming using lasers. So we're also trying to optimize this for other organisms.
Ito po pala question ko, more on behind the scenes 'to kasi the way you explain it
Yeah.
parang ang dali ng ginawa mo. Pero siguro looking back nung ginagawa mo yung PhD, isipin mo walang successful attempt. Tapos ito successfully nagawa mo siya. So ano yung mindset mo nung ginawa mo na like for example, paano kung di ko 'to magawa, eh, di wala akong PhD thesis, hindi ako makakakuha ng PhD.
Exactly. So at that time, because one limitation is if you want to do cryopreservation, you need to have biomaterial, right? So I need to have a larvae. I need to have larva to work on. So I need supply. So when I started, there are times that we cannot get larvae. So that was one of my problems at that time. Because I'm working on a coral called Stylophora pistillata. So it's this coral is a brooder which releases, so it's not like the other corals um that releases sperm and egg. So this coral releases the baby already. So I can get the larvae. But there are times that I cannot get samples because it's too hot. Like there's I think 2020, I forgot what year, but it was so hot. So they are not releasing. So it took me some time to cryopreserve a number of enough larvae for me to write or to have a result. So it was quite challenging and like, can I finish this? Like If I have a lot and it's fine, but like it's not. It's also not that easy
Ah,
to, because it's. I think if I am in the Philippines at that time, maybe because we have more corals in the Philippines compared to Taiwan. But in Taiwan, there are corals, but it's not as many as the one that we have in the Philippines. And I am limited also for the area that where we can get the corals, because I am we can only go to one or two sites in Taiwan. So not like in the Philippines that, oh, if I cannot find it here, I'll go to another place and then you can just get from there. So it's more on logistics, my issue at that time. So how and where can I get my samples? So I did two actually, but the second one, it didn't. They were able. I was successful in cryopreserving them, but they didn't really grow into a coral because that type of coral, it's so it's so choosy with the substrate. So it after thawing, it will just keep swimming. Like at that time, like I don't know which substrate it likes. Because for the stylophora, like it settles on anything. But for the other species, it's so choosy. Like it's been a month and it's still swimming. So it's still alive. It's mobile, but it doesn't want to settle and grow. So yeah, I and then yeah, it's that challenge.
eto po pala, curious din ako sa ano, kasi yung basically yung method nyo, isa siyang way na ma-restore or ma-preserve yung coral reef. Pero may ibang tao na yung research naman nila parang naka-focus sa paggawa ng super species. So, what are your thoughts po sa ganun? Ano yung advantage nitong sa cryopreservation over creation ng super species ng coral niyo?
Um, actually, they're they're two different things. So right now, so during my PhD, I am doing cryopreservation. So freezing what's available. And then your question now is super coral. So now I am working on looking at thermally tolerant corals. So we have a method also now, which is also new. It's called CBAS, Coral Bleaching Acute Stress System. Yeah. So um it's a fairly new technique. It was released 2020. So this method is will identify thermally tolerant corals. So based on this, we can identify which colonies are thermally tolerant to coral bleaching. Especially that we are in the hottest, warmest sea in the world, which is Abu Dhabi. So I think for your question, it's two different things. So freezing is more on preserving the genetic material of the organism. And then if you can pause for a while and where the conditions are not yet okay, and then you will grow them later. So you have this stock. You have this like in the cryobank. But for the super coral, which we don't really like to use, but like yeah, so it's like we this one is we want to produce corals that can resist the temperature, because right now, like everywhere else in the world, like bleaching is going on, like climate change. So we want to find something that can grow in these conditions. So I think you cannot like for me, it's two different techniques. The super coral is where you want to produce something that can withstand heat. But for cryopreservation, it's more on preserving something that's already there. But it's also good to, like, I also want to do that here, cryopreserving the larvae of these thermally tolerant corals, which is
Ah.
good. Because this one is, I don't know, like it's good that I can preserve them, but I'm not sure yet if these corals can withstand heat also. So that's another study. Like it's another experiment that we can do to test the if they are viable for this kind of environment. So they are two different things. So you cannot compare them which one is better or not. So it has its own purpose depending on what question you want to answer.
Balikan ko lang din po pala yung ano, yung methodology and yung result nyo kanina. So basically yung idea, rapidly freeze mo yung larva and then rapidly defrost. Tapos parang tiningnan nyo kung buhay pa yung larva. So curious ba ako gaano kadami actually yung larva na buhay pa after nyo i-defrost?
After ma-defrost, one larva will take me 11 minutes. So in a day,
Tuloy-tuloy
I
na
can
observation yon na 11 minutes, tinitingnan nyo lang siya.
oo, kasi ano kasi siya, it's like, you need to expose remember the cryoprotectant that I told you?
Hmm.
So this cryoprotectant, it needs to be absorbed into the sample, to the larva. So it will take me, so I need to, there's three kinds of solutions that I need to put into the larva so that it will be protected. So one is, so first step is I need to put it like 4 minutes, and then 2 minutes to another one, and then 1 minute to the last one. And then I will freeze them. After freezing, if I want to thaw it, then I will strike it with laser. And then after that, I need to add solution again to hydrate because it was frozen. So it's the water is out. So I need to put another solution again so that it will be hydrated. So if it's just a proof of concept experiment, one larva, that's why it will take me 11 minutes. So 4 minutes, 2 minutes, and then 1 minute, and then plus back again another 2 and 2. So it's so in a day, I can do it depends also on the machine because this is also another challenge when I was doing this. The machine that I'm working with is very should I say this moody. So there are days like, oh my god, please work with me, like please don't. So those are like one of the challenges that I encountered during the actual experiment. So yeah, so your question again is how
Gaano kadami yung nabubuhay na larva after mo i-defrost?
around 60 to 70% survival. It can go also up to like 80 or 90%. Actually, the survival after thawing is high, but the post-settlement survival is very, very low. So settlement is high. It can settle up to like 50, 60%, 70% even. But post-settlement survival is like 5% or even lower. So same as the ones that are happening in the wild right now. Like corals will have a lot of sperm and egg. It will really sperm and egg. There's coral reproduction, there's settlement. But in terms of survival, because that's a different story, when they settled, then there's urchin, there's algae, there's heat, and then so many other factors. So the post-settlement survival is not that high. So it needs a lot of care. So there's a window, maybe first year, that I really needed to do a lot of work in maintaining the tanks. So cleaning the tank, cleaning the algae, making sure that the temperature was okay, the light is okay, the water quality is okay for them to survive the the s- juveniles yeah, juvenile stage, the like the settlement stage. So that's the bottleneck for me. So in terms of survival after cryopreservation is high, but growing them is
Ah,
it's still a work in progress.
ayun. So I guess madami na kaming na-discuss ni Arad tungkol dun sa paper nila, yung results. For anyone interested kung aning yung details sa ginawa nila, check nyo lang ang link sa description. So, ayun, I guess ngayon naman pag-usapan naman natin yung more on behind the scenes naman nung study. So, ito po yung study nyo, ginawa nyo during your PhD. Can you share din po ano yung funding situation nito? Ano yung funding setup? Magkano yung kinailangan nyo na pera to do this at saan siya ginagasto?
Mm, I think I should say I'm pretty lucky in that case because I arrived in Taiwan with um my advisor. He has money. So he will tell me, "Oh, what you tell me what you need and then I will buy for you." So oh, it's so different when I was in the Philippines. No joke. But during my masters, I was a student and an RA. So it's different because I need to work and then I need to go to school. But during my PhD, it's so different because I am a student. Like I think that's why I was able to produce, I think, 5 papers during my PhD because you, I think my task is just you are a student and then you
Ah.
deliver. So my supervisor is like, "Oh, what do you need? You deliver this." So it's a matter of me doing my part. Like I cannot be lazy because it's everything is here. It's kind of like that of a situation. So it was uh, it was a pretty. I think if I didn't have problems with getting samples, I can finish in 3 years. I did mine in like I think maybe 3.5. So because it was also COVID, so I started like a little bit late with the experiment. So it was a good, like I heard a lot of stories, like horror stories during PhD. But I think for me it was good. You also
Ah.
been there, but like it's how should I say that? Like it's hard because you need to deliver, you have exams, you have classes, but I told myself like, "Ara, ginusto mo 'to, wag kang magreklamo." 'Di ba?
Ah.
So and then my supervisor was really really nice. Like he was there. Like if you needed him, like he's there. Like the support was there, which was I am very very grateful for him. Because like so this is I think maybe this one will be good to tell people later if people will want to find an opportunity, a PhD opportunity, an advisor, find really a good advisor. That advisor will fight for you no matter what.
Hmm. May side comment lang pala ako diyan sa sa pag-pursue ng PhD abroad at pag-compare ng experience natin sa Pilipinas lalo na sa UP. Parang
Mm.
pakiramdam ko mas mahirap yung pag-pursue ko ng master sa UP kumpara sa nung nagpe-PhD na ako. Kasi nung nag-PhD ayun nga just as you mentioned nandiyan na lahat ng ng data. Pasok ka lang diyan sa lab office, analyze mo lang. Whereas nung sa Pilipinas ang dami mo pang pagdadaanan na mga fieldwork, mga admin processing, reimbursement na
I
nakakaubos ng
agree.
oras.
I agree. I super agree. I am thankful for all those
Ah.
experiences. I did my master's in 7 years. So in like people now, like I'm working with different nationalities, they will say like, how come? even my professor in Taiwan, like he will not understand why why it will take us so long to finish because we have so many things. But for me, it's also I didn't also regretted doing all those things. Like it was such a nice opportunity. I've been to a lot of places. I've been to like places that people cannot access, but because we are doing research, we can go there. So I never regretted staying in or doing my masters for 7 years. It's fine, but like yeah, so it's really different. I, right? Like
Ah.
I my PhD was like 3.5. My master's was 7. So
Ah. Pero I guess dahil sa mga paghihirap na yun sa ano, nung masters, parang na-breeze through mo na lang yung PhD kasi may malawak na yung ano eh, background mo on how to do research. I guess yun yung malaking advantage nung UP. Pero yun nga
yeah,
lang, yung oras yung ano.
yeah, but yeah, it's I don't know if this is true for you, but if you do your masters in the Philippines in UP and then you go abroad, I think it's easier.
Ah,
Like we have a good um experience in the Philippines, and then going out is it's hard. Maybe it's hard for them. It's still hard, but it's bearable. I don't
ah.
know if you feel also that way, but like yeah, you will still not sleep, you will have long hours, you need to study, but I don't know. It's it's
Ah,
bearable.
gets ko yung gets ko yung feeling na yan.
Bearable. It's okay.
Ah.
Yeah.
Dun po sa ano, sa during your experiments, like can you share din po yung moment na ano, nakita niyo na successful yung experiment and na-realize niyo na ano, yun nga may breakthrough na nangyari. Nakagawa kayo ng breakthrough.
Uh, I because like what I've said, it's a very young science. So when it was starting to grow, like in like even 1 month after it settled, like oh my god, you're alive. You know that feeling, like oh you're alive, and then it continued 2 months. So it was that time, like when I was able to even like when I was able to produce a coral spat as a coral that settled for a month was already a success for me. And then I have another paper that I looked at the ultrastructure, the internal, like how the ultrastructure or the structure of the larvae after I used transmission electron microscope. So I looked at how the changes of the internal structure of the larvae after cryopreservation. So that one, it gave me more confidence why my larva settled because of these factors. But this is a different paper that I published on coral reefs. So there's damage, obviously, because you freeze them and then you hit them with a laser. There's damage that you can see in the cells, but that damage was not that bad for them to settle. So that paper backed up my findings for the settlement of the cryopreserved one. So I was like, okay, I'm convinced now that it's good. So because it's not just the larva that you can see, but it also backed with um, a microscope technique to see what's going on inside the larva.
Ayun, so nakakaisang oras na tayo magra-wrap up na po tayo.
Oh,
So ayun, before
yes.
we let you go, Ara, ano ang mga advice mo sa mga aspiring researchers?
I remember when that time when I was about to start my PhD. So I messaged my professors, so Dr. Alino, Dr. Liquanan, and other people from the Philippines. So they told me that because I told them like I'm starting my PhD, like I want I hope the stars will align for me. And then they told me that, oh, you should work. I think you work hard or even harder than you are doing right now. So I think for me is like we just work and do what you do best, but like also don't forget your mental. That's also important.
Mm.
Like it's not purely work. Like for me, I enjoyed, like I do a lot of like hiking and swimming and doing non-scientific things, but also do like when it's time for experiment, when it's time for work, I work. And then when it's not, I also do relax. So a balance of, and I think it's also good like to try things because you'll never know. Like for me, my background is more coral ecology, and transitioned to cryopreservation that I don't know anything about. But I know I have my background about corals and coral biology, but the technique was so new for me at that time. I don't know anything about it, but I just tried, and then it worked out for me. So I think, and also it's different. I'm not it This is very I think it depends which country you came from, but it's like for me, if there's an opportunity, you grab it because there's not you don't know if there will be another opportunity for you. So if you're ready, and then you just grab it. And then because like I said, I'm not really a planner, so I work whatever is there, not too far from the future, but like so whatever is like if there's available opportunity that you are also like, oh, I can do this, I want to try this, then maybe grab that opportunity. It might take you somewhere. Yeah. And I think also enjoy. I think also is like enjoy like the things that, or like the field that that person wants to be in, is also a matter of I like this job or I like this field, because it will give you another like, oh, if it's hard but I still like it, so it's okay, right? Like I enjoy doing this even though
Ah,
it's hard. Like it's impossible to find a work or a job that will not require stress.
ah,
Stress is always there. But if you love that thing that you're doing, stress will be bearable. Like you will not sleep. Y- you will have a lot of experiments. You will have a lot of field work, or you will go to places where it's so remote, but it's hard, but at the same time, you like what you're doing. You want to do this. So it's okay at the
mm.
end of the day.
Ayun. And last question po, do you have anything to promote?
To promote, like, okay, so right now, so from cryopreservation, I am transitioning, not really transitioning, but I'm adding something. So I'm currently working on thermal tolerance of corals. So we are looking at colonies that are thermally tolerant to stress, to heat stress. So we are exposing corals to 40 degrees of, so it's very hot here. So 42 degrees of water temperature. So um hopefully maybe next time if I will have a chance to talk with you again, I can
Mm.
share like what we are doing in the Gulf. So the Arabian Gulf, or like where we are right now, I'm in the UAE. It's we still have corals in this region. So the lessons that I might, or the techniques that will learn from here, because it's still starting our project, I can maybe bring to the Philippines later. Like maybe we can choose also corals that are thermally tolerant, and then with the bleaching that's been going on and the frequency that has been hitting the reefs all over the world, will also help. So will also help us here, but also that we can also bring, that I can also bring to the Philippines later. So we'll see.
Nakita ko sa balita parang a few months ago, mag-e-establish na rin yata ng first cryo bank for coral
Yes.
larva sa Pilipinas.
Yes. So that was my lab before. So if I didn't come here, I might be there also. So,
Balik
but like,
scientist.
yeah, there, balik scientists. We'll see. We'll see. We'll see. We'll see. Yes, it's a very good. Like, this is a very good project too that my supervisor before is spearheading. So there's different countries that's involved. So there's Philippines, there's Malaysia, Indonesia, and I think Thailand. So Philippines is it. Yeah, we will have the first. I think first. Yeah, it will be the first in the Philippines. So it's a good one. So maybe there's also like from my lab Filipinos who are in the lab doing their PhD now. So we'll bring this back later in the Philippines, hopefully. So what I hope like later what we are doing also in wherever we are, not just me, somehow we can bring it back to the Philippines and apply it in the Philippines so that we can also benefit from what we got from other countries.
Yes. Ayun, so I guess hanggang dito na lang muna tayo. Maraming salamat kay Ara sa pagpapaunlak ng aming imbitasyon. At maraming salamat sa lahat ng nakinig at nanood. See you everyone next week. Madami pa tayong pag-uusapan na research na ginagawa ng ating mga Pinoy scientists.
Thank you, Paul.
The opinions of podcast creators, hosts, and guests are not necessarily reflective of the official stance of the Pod Network Entertainment, its hosts, or other network programs. The content created by the people behind the podcast is personal and not meant to harm any religion, ethnicity, group, organization, company, or individual.